Hello, everyone. I hope that no one is falling asleep at this time of the day. And first of all, I would like to thank Steve and Victor for asking us to do a sponsorship for this event. It's funny because I was not supposed to be here, neither any of my colleagues, because today was supposed to be in Valencia. But as you know, I couldn't be there, and they told us actually yesterday. So I called Steve, well, I sent him an email like, hey, I can make it to the event. Can you give me 5 minutes? And I was ready to negotiate. I would be down to 2. He gave me 15. So that was more than enough. I would really like just to introduce a little bit who we are, what we do. So if it's interest for you in the future, but I'm quite sure that at least most of you are going to know Waters. It's a big multinational company that they do HPLCs, the mass spectra basically. And I am in the TA Instruments division. It's a division more focused on materials, you would say. The TA actually stands for thermal analysis when they started in DuPont in 1964, if I remember well. So we have a couple of corporate slides. I'm going to go through them quite fast because I don't think it's really necessary for you to know all the story. We have a vision, we have a mission too. We're a big company and we're here to try to help to solve problems. We do have a lot of different instruments and we are a really, really big company. 2022 revenue was close to 3 billion. We are more than 8,000 people in the whole world. Here in Spain for Waters, we are close to 100. My division is a bit smaller, like a 10% of that, but I like to think that we do our work well, so it's efficient. And we do have 2 laboratories in Spain, actually, one in Madrid and the other one in Barcelona. That would be here, you can see, and also another one in Portugal. So, as I said before, Waters is mainly known for liquid chromatography. We would be in this area here that is called materials characterization. And materials characterization, it's a very weird word because it involves a lot of things. We could be thinking about, for example, polymers. It's one of our biggest markets. Batteries, automation, petrol, cosmetics, food, and also pharmaceuticals. That could be both small molecule or big molecule. For example, ibuprofen, or we can be looking at proteins or antibodies, for example. In this case, for this talk, we are looking only at life sciences for testing pharma and biopharmaceutical drug efficiency. We have some instruments that I think that could be, I mean, you could need them at some point, and if not, if you don't want to buy one for yourself because you think that you're not going to be using them that much, Solitek, they have DSC and TGA, and I know that they use them a lot. We started working with them since the beginning. This is a quick picture of the portfolio that we have at TA Instruments. This is not Waters. This would be only our division. The instruments that we are going to be talking today for some minutes are basically thermal analysis that would be involving DSC, calorimetry, and TGA, that's thermogravimetric analysis. And then we're going to be focusing a little bit on the microcalorimetry. There is also a part that could be interesting, biology, but there is not enough time to talk about that. So if anyone has questions, please let me know at the end. Let's start about thermal analysis. And I'm going to apologize because some of these presentations are in Spanish because I was sick when I was doing it, and my head was not straight at the time. But I think it's quite easy to understand. This is thermal analysis, starting by DSC. This is differential scanning calorimetry. You put a sample in here, you apply a speed rate, a ramp of temperature, and you look at all the transitions that you can find in a molecule, like fusion, for example, crystallization. You could be looking at a phase change. Glass transition is also very interesting for amorphous materials. This would be for some of the things you can look. So amorphous materials, polyformism, compatibility with other molecules, and glass transition or freeze-dried materials. Not lyophilized, freeze-dried materials. One of the very common applications would be this one, is that you take one molecule and you set it up with other, for example, this aspirin with magnesium stearate, and when you put them together, you can see that instead of looking at the fusion of both materials, you're looking at a completely different thing. That means that these materials are not compatible. They are going to decompose with time. Just so you can know it, I mean, we do have one instrument that can do up to 3 samples at the same time. If you have some quality control issues and you need to do a lot of samples, that could be a good option. And also, I don't know if the technology is going to be the same as yours, but we do have several cooling systems that they don't work with liquid nitrogen that could go down to -120. Those are compressors. I'm pretty sure that before was a Peltier system. Before was a Peltier system, the one for the polar bear. No? Oh. Can we talk later? I'm interested in that. But well, we can reach quite low temperatures without the use of liquid nitrogen. That's very convenient because liquid nitrogen, it's always free for the universities, but in a private company, it's a pain in the ass. Trying to buy it and they send it to you every week. Well, we know the perks. Then for the TGA, we are looking here at a thermal balance. So we have here on top a balance, and then we have here small hang down. You get the sample on a small capsule, and you weight it. And you weight it through the whole range of temperature. You start applying a scan rate, and you look at all the different losses of mass that you can have in a molecule. That could be, for example, just because of the degradation, you have a molecule that is decomposing with time and temperature. It could be because you have some volatiles like water or solvents, and also you can connect this instrument directly to an FTIR or mass spectra. So the composition of these materials can be analyzed by the FTIR or mass to exactly know what you had before. And that also could work for the solvents. If you have water, you have some solvents from your reaction, I don't know, DCM, THF, ethyl acetate, you can actually analyze that and to know exactly the proportion that you have in the sample. This is one of the graphics that you normally see. You only look at weight and temperature, and you can see you have here different losses. We always look at the derivative because it's easier to take a look at that than just looking at the small degradations here, because you have peaks when you look at the derivatives. And you can integrate those and to know exactly what is the percentage and at what temperature they happen. Just a quick look at a couple of more instruments here. We have the high pressure TGA. I don't think that that would be interesting for your markets, but I think it's just wise to show you what type of instrument it is. This is a TGA, but it's inside of a magnetic balance, so it allows you to work up to 80 bars. For some people, they bought this instrument because it looked like a really, really small reactor too, because you can work also with solvents and with vapor too. And the second one would be the Discovery VSA, and I do really think that this one is really, really interesting for the pharmaceutical industry. This is a TGA that can apply humidity, and we talk about humidity, it's humidity, not vapor. We look at a very narrow range. It works between 4 or 5 degrees up to 95, if I remember well. But it can work, 85, it's just down here. And it can work from 0% to 100% of humidity to study how humidity would affect your molecules. Are those molecules recrystallizing because of humidity? Are just hygroscopic molecules? And those hygroscopic molecules can get the water and then release it, or is it going to stay there forever? These are a couple of the experiments that you would get for hygroscopicity, and then this one would be for a change of phase from an amorphous material to a crystalline material. There is a point of no return that when you go sometime, in some molecules, of course, not all of them, when you go over that humidity and the temperature, the molecule would recrystallize and it would change the polymorph from amorph to one polymorph, or from polymorph 1 to polymorph 2, for example. And this is very important for storage. Because if you get something at 40 degrees at 20% humidity, what's going to happen after 3 or 4 days, for example? And then, just getting to the last part of it, I think I still have 5 minutes, yeah? We have the microcalorimetry section. This is a completely different one. We do have some people here that they have this instrument, and it's different from the DSC because this always works at an isothermal temperature, so always keeping the temperature constant. And the idea is to see what is happening to your molecule. Is it going to be stable? It's not going to be stable. You can add some vapor if you want. You can try to even replicate what it would be the dissolution of a molecule. This system here is the Solcal, and you get your sample is inside of a small crushing ampoule made of glass, and basically when it starts, it hits the bottom, it releases your molecule and starts dissolving it. And you can study the dissolution of your material and study the enthalpy, how much heat does it generate. That could be both used by research and development, but also for quality control, because if one of your samples, it doesn't have the correct crystalline structure, this would look different at the end. It would take more time, it probably would take more heat or less heat. And of course, here the other one would be isothermal calorimetry, just looking at keeping the instrument at one temperature and see how it affects with time. See the heat flow of your sample. The idea would be that it should stay always at 0, but nothing stays at 0. So it's going to be always a bit up, and then you can check if this is inside of your parameters or not for quality control or R&D. And the last but not the least, inside of our microcalorimetry section, we also have a couple of more instruments that I do think they are interesting, but I am not sure that there's many people here working in biologics. Is there people working in biologics here? No, right? I'm just going to talk for a minute about that. These instruments would be the Nano DSC and also the Nano DSC are really focused on the study of the interactions between a ligand and a protein. That's why I was talking about biologics. This instrument, you can use it for a lot of things too, because at the end, it's like an automatic titrator. You can get a protein at the bottom, you can get just a molecule that is going to interact with something. You can add a ligand that could be a small molecule, you could add a metal if you want, and it's going to give you a graphic when you're going to have at the end a constant of the affinity between your ligand and your sample. And you can also get a model, get all the kinetics that you need on your molecule. And the last one would be the Nano DSC. This is a DSC. It's a DSC like the first one, but this one is focused on proteins. And why do we have to separate from the classical DSC that we can work with solid samples like a molecule, like aspirin, for example, or with a polymer? We should switch to this different instrument. It's because of the sensibility. Normally with proteins or antibodies, you need to work at very, very high dilutions, and we are looking at nano transitions. With DSCs, we're looking more at the millibatt transition. It's just going a little bit below the limit of the instrument. And we would look at protein stability, structure, and ligand binding too, for example. And just, I promise that this is the last one about the instruments. We just released, beginning of this year, a new instrument that it's called the Rapid Scan DSC, the RS DSC. This is a Nano DSC too. But it's a Nano DSC that offers two different things compared to the other one. High throughput, you can get up to 24 samples each time, and also, it's not, yes, here, sorry, and strength, you can actually work with very high concentrations. Normally when you work with a Nano DSC, you would be looking at 10 to -4, -5, -6, smaller, but in this case, you can go up to 300 milligrams per milliliter. And as far as we know, in water state instruments, there's no one that can do this right now. And we started developing this with the pharma industry in the United States because there was a need to study high concentrations, because most of vaccines, as far as I know, they work at high concentrations, and the behavior between one sample at 10 to -4, -5, and 300 milligrams per milliliter is going to be far higher. It's completely different. Well, yes, just to finish, here in Spain, Victor and Steve, they know us. I mean, I'm the sales guy, so I'm the bad guy, but we do have some people from applications. We have technical service too. Those people are to help always our customers, and we have a really, really good application support. When a customer of ours, they have a doubt, they can just write us an email, we can call them, and we try to help us as much as we can. That's the main idea, not just selling an instrument, we're selling a technique. And if there is doubts, you can always come back to us to try to see if we can help you with that. Because sometimes you're the ones that know more about your samples than we do, but we are always in there to help. And just to finish, there in the website, I think that this could be interesting. If you go to the applications part here, there is one page for pharmaceuticals, another one for biologics. There is information about the instruments and the techniques, of course, but there's also a lot of webinars, even a couple of ebooks that I think that would be interesting for you, that they talk about what we do, of course, and how you can apply it in your different fields. And this one would be the one for biologics. But you go directly to tainstruments.com, applications, and you can go now to the different sections. So, thank you very much. Is there any questions?
I think we got time for one question, perhaps. Okay. I'm going to ask you about the Nano DSC that measures the interactions between the protein and the ligand. Do you think that, obviously, as a screening tool, could it be used in discovery teams to determine the activity of the new compounds that they are making, let's say, in parallel? Or you know, if you're making enzyme inhibitors, for instance, where you're measuring the affinity of the substrate or the enzyme for the substrate, could you use it as a tool at that stage and skip the traditional in vitro activity testing?
Yes, yes. Thank you very much, Victor. It's a really good point. And perhaps I should have stopped here for 10 more seconds at least. This is a really good tool for that, because we are looking at the affinity between two different molecules. These molecules could be big molecules, could be small molecules. I use that instrument to check how my porphyrin derivatives, that there's molecules at 500 grams per mole with metals, but you can put there a protein, you can even put a receptor on top and you can add it, and you can look at the affinity between those. So before going directly to in vivo, this would be a faster tool to see if there is an affinity or not. Of course, going into in vivo, it's going to be a lot of more things to take into account, like the cell is going to be alive. I mean, a lot of more things, the distribution of the drug, but most of the pharma industries right now are using this just to see the affinity of the new drugs with receptors. It's a well-known, and you can call it a gold standard right now.
Okay, thank you very much.
Thanks.